This is going to be a little bit of a different flavor of a lecture. The extreme part is I'd like you to picture doing what I talk about in your kitchen within the next 5 years, because I think that's where this technology is going. And I've got a little demonstration that's running up here that we'll talk about towards the end of my talk. So what really excited me getting into this field was I did a master's degree down in Virginia in biogeochemistry, and we talked about microbes as just sort of this amorphous black box of biology that we didn't really know anything about them. So this was in the late 90s. And then for my PhD, I moved out to California, to Los Angeles, USC, as Darlene mentioned, to join a lab that was using some of these new cutting-edge molecular-based techniques where we could start to actually dissect these microbial communities using DNA-based technology. So the organisms that we typically work with are divided up into two major groupings. There's the prokaryotes on the left and the eukaryotes on the right. So the eukaryotes are all the organisms that are featured in Tiny Giants, microalgae, the protozoa, things that look like paramecium that are eating biomass in the oceans. The prokaryotes doesn't really do this group justice because it's actually split up into the archaea and the bacteria, and the archaea are just as different from the bacteria as the microbial eukaryotes are. So I'd just like you to keep these organisms in mind as we go through this talk. So again, I'd like you to think about this as a tutorial on an area of technology that just turned 30. And so you may or may not have heard about it. And I'm going to be building up to actually revealing what that technology is and what the technology can do. It's been used almost exclusively in research labs and clinical settings for the last 30 years, and it's really changed the field of biology. It's allowed unprecedented access into this unseen microbial world. I'd like to make a prediction here that you or your kids or your grandkids will probably be adopting this technology within the next 5 years. It's actually already starting to trickle down into general biology curriculum. I also predict that this technology will have profound impacts on many areas of our daily life, human and ecosystem health, ecology. We use it for ecology a lot in oceanographic research, conservation biology, agriculture. We're also actively pitching the technology to people that are involved in the aquaculture industry, which is a new and emerging industry in Maine that's really poised to take off. And then just general scientific curiosity. I think there's a huge application for people that just kind of want to know more about the natural world. So I look at this technology that's coming to everyday life as a democratization of access to the information that's stored in every cell of your body and in every cell in the world around you. There's a huge amount of biological information that we generally just walk past every day and don't really think about too much. So an overview of the talk. I'll give you a primer into the science behind DNA technology just to get everybody up to the same page. We'll actually look at the technology pretty closely that I'm raving about here. Look at the history briefly of the technology and a couple of the different flavors of it. And then we'll talk about some of the applications of the technology in research and increasingly in everyday life. And then I'll finish with a brief demonstration. And as Darlene mentioned, please just jump up and down, shout out questions, whatever you like. So I'm talking about DNA coming out of the lab and into your everyday life. DNA is organized in chromosomes. The chromosomes can be broken down into chromatin. The DNA sequence itself is wrapped around histones. This is in most eukaryotes. Bacteria lack the histone organization. And then ultimately, if you look at a very fine level of detail, you've got the double helix, which most people have probably heard about, and then breaking it down to the single strand. One strand is the top strand, or the coding, the sense strand of the DNA, and then one is the anti-coding strand, or the opposite strand. And so this is basically the basic structure of DNA in every living cell for a eukaryotic model. The central dogma of biology says that information is stored in DNA. DNA can be replicated within the cell. It's then transcribed to produce a message within the cell. And the message is in the form of RNA. The RNA is what actually gets translated by the cell machinery into proteins. So it goes from the potential of the cell to the message to the actual function, because it's actually the proteins that we rely on for performing necessary cellular processes. And then what this might look like inside of a cell: double-stranded DNA, single-stranded mRNA messages, and the messages get interpreted on ribosomes that ultimately synthesize proteins, polypeptides. Just another way of looking at it. When it gets transcribed, the T's, so DNA is made up of 4 base pairs, okay, nucleotides. It's the A, C, T, and G. So that in the message, those T's are converted to U's. It's just a slightly different chemical molecule. And then it's actually the order of these triplet codons, UGG, that code for specific amino acids that make up proteins. So when we're presented with a genome, which is just a huge long string of A's, T's, C's, and G's that look sort of like they're in random order, we can start decoding it using an amino acid codon table, or a wheel, as in this case. And the codons are read in this order, and they code for all 20 amino acids, essential amino acids. So for example, the first position would be a G, second position a C, and then you could either have a U, C, A, or a G, and it would code for this particular amino acid, alanine. And so the codons have some degeneracy built into them, and that's to give some flexibility to the cellular machinery. When you're looking at a genome, you want to actually find the genes, the bits of the genome that actually have the information. And to do that, you look for the codons for the start. So at the beginning of every gene starts with an AUG. And this is a start codon. It also codes for methionine. There's a couple of different codons that will actually tell you to stop. And so that's typically the end of a gene. So the next time you download a genome off the internet, this is the genome of E. coli. And so it's a pretty small one. It's about 4.6 million base pairs. And this is about just 0.15% of it. It's all I could really fit on the screen. It's 6,800 base pairs. But now you know how to look through an E. coli genome and figure out where the genes start and stop. And based on that, you could actually just use a text editor and cut out little chunks of that genome and then send it into internet-based searches, and it would pop back an answer of what gene you're actually looking at. So it's really not as scary as it sounds, and it's a little overwhelming to look at thousands and thousands of individual nucleotides, but it's within your power to start analyzing genomes. Okay, this is fine for E. coli. It's only 6,800 base pairs, not too bad. Again, just these open reading frames are the putative genes, and an open reading frame is sort of a proxy name for a gene, but we don't necessarily know if it's a coding gene or not. It starts with the methionine, the start codon, AUG. In this particular example, it ends with the UAA. Okay, so now you know how to predict amino acids from DNA sequence. And with amino acids, you can build proteins. So one thing that's really interesting about genes in genomes is that they're not just straight words. You can't just read through a genome and necessarily find the exact, complete mRNA message, because they're interspersed. So there are coding regions called exons, which ultimately become part of the message. But they're broken up, they're interspersed. The different exons are interspersed throughout the genomes. And so you'd have a start and a stop, and then you'd have this non-coding region, and then a start and a stop. Another non-coding region. And the reason for this is that it allows for alternative splicing. So with this same set of nucleotides, you can come up with several different proteins just by rearranging the exons in different order, or not putting them all together into the same message. So one way I like to think about this, I was trying to think about how to explain this, is that if you had imagine the longest Harry Potter book, which is, I think it's about, 200,000 words, but imagine there were no spaces in between all the different words, and you were told that you had to write a new book using all the words in this very long book. Well, you wouldn't really have any choices if the words were all crammed together. You'd simply have to copy that message. But if you could shake those words out of that book and then rearrange them, you could probably write a complete new story. And that's sort of how you should be thinking about the messages that are interspersed throughout a genome. Okay, so I gave you an example of a genome that's fairly manageable from E. coli. Now, humans have haploid genomes of about 3.3 billion base pairs. Okay, only about 1.5% of those base pairs code for genes that have a known function. There are many other regulatory regions in genes that just help basically create the messages, make sure everything is done in the correct order. But our genomes are minute compared to organisms like this. This is a dinoflagellate, so it's a cousin of the red tides that we have here in Maine. It's a cell that's about 10 microns across, so about 10% of the width of a human hair. Its genome is approximately 20 billion base pairs. Nobody has sequenced a complete dinoflagellate genome, and even if they did, I would predict that greater than 99% of it would be, we wouldn't know the proteins that it actually coded for. So it's a really interesting model organism, and there's a lot of research right now going into figuring out why dinoflagellates have such immense genomes. Okay, so accessing genomic information. Genomes contain a wealth of information, as I think I've told you already, about the organism's functional role in the environment, its health, its evolutionary history, and its ecology. So there's a huge amount of information embedded in every cell of every organism. So we're getting close to the point where it's becoming practical to sequence an entire genome. And in fact, for a bacterium, it can be done very quickly. Whenever there's an outbreak of a foodborne pathogen, typically the genome of the offending organism is found within a matter of weeks. And finding it is the hard part. Sequencing its genome can be done in a single afternoon. So that's a technology that we have at the lab now. Not that we're doing pathogen detection, but we have the ability to put together bacterial-sized genomes within a period of a couple of days. But what if we could reach into a genome and pluck out particular genes of interest that might tell us something about the taxonomy or the function of the organism? And we could do this for any single organism on Earth, a bacterium, an archaeon, a protozoan, a plant, animal, fungus, virus. What if we had a process that could do that? Luckily we do, and that's why I'm here tonight. So I'm going to tell you about the polymerase chain reaction. You may have heard about this, read about it. It's made it into the popular press. It was invented, I say mostly, in 1983. There was some initial research that was done back in the early 70s that kind of described a similar process, but nobody really put it all together until the story is that Kary Mullis, who was a biotechnologist at one of the first biotech companies, Cetus Corporation, was driving down coastal Route 1 in California one night, and it suddenly popped into his head about how to solve this problem of amplifying whatever DNA sequence you wanted out of a genome and then being able to analyze it. And he got, for this development, he and Cetus Corporation shared the Nobel Prize in Chemistry in 1993. And one of the big advances was that biologists already knew how to copy DNA, but the problem was that the DNA polymerase, which is what assembles the individual A's and C's and T's and G's, wasn't heat stable. So you'd add DNA polymerase to a reaction, and then it would break down, and then you'd have to basically go back to the lab bench, add more, and then shuttle. So it was basically a lot of manual, hands-on shuttling of reagents around, and it was very costly. Well, one of the big breakthroughs was when Kary and folks at Cetus isolated a DNA polymerase from the thermal hot springs in Yellowstone. It's called Tack Dna Polymerase, and it's heat stable. So it can take being heated to 95 degrees Celsius 40 or 50 times and still maintain its function. Because of course it comes from an organism that's practically living in boiling water. A different company, Perkin-Elmer, developed an automated approach for amplifying DNA with a small machine, a bench-sized machine. And this really was also a significant breakthrough in the whole process of being able to set up a reaction, put it in a machine, press a button, walk away, go do something else, and you come back a few hours later and your reaction is done. PCR has been incredibly valuable. It was sold in 1991 for $300 million to Roche, Hoffmann-La Roche. So it's been had a huge impact in the biotech world. If you want to look up the first paper that was published using this process, it was in Science in 1985 by Sake et al. Okay, so what is PCR? PCR is an amplification process. You know a little bit about a gene of interest within a particular genome, or maybe it's just the entire environment. You collect a soil sample from your garden. You collect a water sample or a sand sample from the beach. And all of these things are real-world applications of the technology. You know a little bit about the gene of interest. You add all the different components, the A's, the C's, and the T's, and the G's. Those are depicted by these little blue shapes. You add the DNA primers, which are just short, single-stranded DNA molecules that help kickstart the reaction. And then you heat it up. You drop the temperature a little bit so that the primers can come in and find their complementary positions in the genome. And then you heat it up to the optimal temperature for the DNA polymerase, which is 72 degrees Celsius. So picture going from 95 down to somewhere in the 50 degrees Celsius range, and then back up to 72 degrees. And you do that over and over and over again. And every time you go through one of those cycles, you double the amount of target DNA. Okay, so it's an exponential reaction. So very quickly, you build up thousands and thousands of copies of a gene.
And nothing is killed.
No, there's nothing living in it per se. You've got a functional enzyme, but otherwise there's nothing living in the reaction. So what does it look like?
Excuse me?
Yeah, go ahead.
What did the 3 and 5 stand for on the previous one?
Oh, so that's actually just means It's the directionality of the DNA. DNA is always synthesized from 3' end towards the 5' end. So you can see here that the red, again, are these short little priming sequences that come in and find their complementary strand on, in this case, the top template, and in this case, the bottom template. The DNA polymerase comes in, finds that interface between single and double-stranded, and then extends. It incorporates all of these nucleotides into the correct order based on what the nucleotide is on the top strand. Okay? So it always synthesizes from the 3' end of the template towards the 5' end. It's just a road marker, or road map, basically. It's a good question. So typically, we just set up these reactions in the lab, and we let them react for 20 minutes. We walk away, we go get a cup of coffee and catch up on our lab notes or something. And then we come back, and we have to have some way of visualizing the DNA that we just amplified. So typically what we do is we load it onto an Agarose Gel. So Agarose, it's basically it's sort of a jello-like material, comes from kelp. There's actually a company here in Maine that produces very high-quality agarose that we all use. This happens to be a prepackaged gel, and so we can just, as soon as the reaction's done, we take the tubes out of the thermal cycler, and then we use a small device, a pipetting device, to load a little bit of the reaction into a tiny chamber at one end of the gel. So the gel is actually submerged in a conductive buffer. We turn the electricity on, and the DNA migrates from the negative pole towards the positive pole. And so we run that for about an hour, 45 minutes to an hour, and we always load a DNA ladder. So this is just a size standard here. And we typically know how big of a PCR product we should expect. And so we come back, and these gels actually have a DNA binding stain right in them. So we can, as soon as the gel is done, we can pop it under blue light, we turn the lights on, and then these bands start glowing. And then we just use a typical digital camera, snap a picture, and there are our PCR products. Okay, so we've just fished out a gene of interest from the environment or from a culture of a particular organism. So after that, what can we do? Well, we literally just we crack the gel open, we can cut these bands out, we purify them and concentrate them, and then you could actually do direct sequencing on those PCR products if it was from a single organism. But at Bigelow, we tend to work out in the environment, and there's thousands and thousands of organisms all mixed together. So a band like this might represent the DNA from hundreds, if not thousands, of different organisms. And so to actually sort those out, we have to put the PCR products into the bacterium E. coli. We plate out the E. coli, and then we pick individual colonies of E. coli, and we let that bacteria actually replicate the DNA for us a second time. And they make almost exact copies of the DNA, and then we can actually just sequence the gene back out of the bacterium. So it's kind of a way of sorting out all the microorganisms in the environment. Now, the way DNA technology is going now, you could actually just take a PCR product like this, and using some new molecular techniques, you could get individual sequences for each one of those DNA fragments that makes up each one of those bands. Still have everybody here?
Sort of.
Sort of, okay. Slow me down or stop me, ask those questions. Okay, so that was one flavor of PCR. The other flavor that I'm really excited about, and we use a lot in my lab, and my students are available to answer all kinds of questions about this after the talk, it's called real-time quantitative PCR. So in quantitative PCR, after every single cycle of the amplification, we take a picture of the reaction. There's a fluorescent molecule in the reaction that reports to us at the end of every cycle. And remember, I told you that it's an exponential process, so that's what's described in this graphic here. At the very beginning, the first 10 cycles or so, there's really not much. There's amplification going on, but you're going from 2 to 4 to 8 to 16 to 32. Eventually, you start really cranking up the doubling of gene target. And so this is a typical amplification profile. And the only reason it stops is because at some point you start to run out of reagents. Okay, so eventually it hits a plateau. And the important piece of information here is the cycle number. And again, this is just about 1 through about 30 or 40 different cycles that you run that 95, 55, 72 over and over again. It's the cycle number at which it crosses some threshold that's drawn by the software that's part of the PCR machine. Okay, so the piece of data that you're recording, we don't care how high it goes, we only care where it crosses the threshold. And we can run reactions like this, and then we can also run, so if these are unknown samples that we don't have any idea how much of the gene was in the initial starting sample, we standardize it by running known copy number of different reactions with different amounts of starting material in them. Okay, so you could picture a graph like this, but then with evenly spaced amplification curves that were due to the standards. So then, if you take the information, the cycle number at which amplification crosses a threshold, which we just get by dropping down from the point where it crosses this dotted yellow line, and then we plot that against the copy number in our standards, we can then relate our unknowns to our standards and figure out how much DNA was in that initial sample to begin with. Okay, and by knowing something about the copy number of a gene in a particular organism, you can figure out how much of that organism was there to begin with. Okay.
Do you determine the threshold? Or
there's some complex math that goes into that. I usually let the machine determine the thresholding. But it's basically, it's an area in the exponential phase of the amplification, but not too far up, if that makes sense. So these are some real data from an assay that I developed when I was in grad school, and it shows this amplification from just 6.5 copies of the gene in a little tube to 6.5 billion copies. And it's a linear process. You can draw a straight line through those data points over, what is this, 8, 9 orders of magnitude. So it's incredibly sensitive, and this is where it becomes very useful for diagnostics, in particular medical diagnostics. If you get bit by a tick, this is the kind of test that they will do to figure out if you have Lyme disease. Okay, it's a PCR-based test. It's also, when my family and I lived out in Los Angeles, we all got swine flu that year. It was, I think, 2009. And I was on the CDC website looking up the primers for that particular virus because I was close to we had a 1-year-old daughter at that point, and the LA Health Department wasn't testing people anymore. But, as a scientist, I really wanted to know. So I was really close to buying the primers for 2009 swine flu and just running the test when my advisor wasn't looking. So what are some of the applications? We use it a lot in microbial ecology for counting specific microorganisms. You've probably heard, if you've been to any Bigelow talks, that most of what grows out in the ocean can't be grown in the lab. And so we want to know something about how many of those organisms there are. It's really useful for pathogen or parasite or harmful algae detection. Jose, who's in the back, and I just got some funding to do a lot of PCR, quantitative PCR, for some of the parasites of oysters that he's an expert on. So we'll be starting that project probably in the spring of next year. It's incredibly useful for detecting bacterial or viral infections, in human health, aquaculture, agriculture, pet health, right? Your pets get sick, they can't tell you how they feel. Do a PCR, figure out what's ailing them. Genotyping, it's really useful. It's an incredibly sensitive technique. You can detect, there are methods that can be used to detect just single base pair differences in particular parts of your genome. And increasingly, we're finding out that these single base pair polymorphisms, they're called SNPs, single nucleotide polymorphisms, are really important in determining disease or predisposition to particular diseases throughout your life. So it's incredibly helpful in improving our health. And then we're also using it for studying gene expression. So one thing that I didn't tell you about is that you can capture the message in between the gene and the protein. So we extract the RNA, we convert it back to DNA, and then we can relate the amount of message to a particular environmental condition. So we can figure out how these organisms are making a living in the environment and what's causing them to turn on particular genes at a particular time or shut them off, as the case may be. So over the last 10 or 15 years, I've used this technology to target a bunch of organisms that are interesting ecologically, including this marine algae Phaeocystis. It's found in incredible densities down in Antarctica. It's globally distributed. We have it out here periodically, usually in the very early spring. But each one of these little tiny dots is a single cell, but at some point they all come together and form these beautiful colonies. So we don't know a whole lot about its biology when they're single cells, but PCR can tell us how abundant they are throughout the whole year. The first assay I developed was for the world's smallest free-living microbial eukaryote, Austriococcus. It's the size of a bacterium, but it's a eukaryote, meaning that it has a membrane-bound nucleus. So it's one of our very, 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 very distant cousins, but it's a eukaryote like we are. And then it's also really useful for monitoring the abundance of harmful algae. So that's another area of interest of mine. And we've been working for the past couple of years on detecting this organism, Pseudonychia, which produces a very potent neurotoxin and is found in local waters here. Luckily at fairly low abundance. And then when we compared a PCR-based method to a more traditional method like flow cytometry, which is another method, if you come on Friday, you'll learn all about flow cytometry. And the blue line here is a plot of Austriococcus, or what we think was probably Austriococcus on flow cytometry. And then the green line was my gene-based detection method. So you can see there's a very high correspondence between the two numbers. The other thing that it can be used for in the environment is this genotyping. So in this little study, I took 22 strains of the red tide algae Alexandrium from the culture collection that we have at Bigelow. I amplified them with a set of PCR primers, which are specific for that genus of marine algae. And then at the end of that amplification process, I melted all the DNA. So I stepped up the temperature of that reaction very slowly by 0.5 degree every 5 seconds. And then I took a picture of the reaction. And when you do that, you can capture how the DNA falls apart. So you've just amplified a whole lot of DNA, and now you're cooking it, and that double-stranded DNA is falling apart. And when it falls apart, it's no longer fluorescent. Okay? And the rate at which it falls apart can sort out different species of marine algae. And so that's what we have here. So we've sorted out all of these algae without even accessing their genomes. We've just done it sort of using this proxy method, amplifying one little target region of their DNA. And one of the reasons that I did this is that we thought we had a misidentification of one of the algae in the culture collection, and it turned out we did. It was labeled as Aphinae, and it actually turned out to be a tamarance by this analysis. So we could correct these errors. But for diagnostics, it's just an example of showing you how you could use it for genotyping and rapid diagnostics. And then most recently, we're trying to use the technology to link it up with studies of biogeochemical cycles in the context of ocean acidification. So we're using the giant ocean mesocosm tanks. We just did a pilot test over the last couple of weeks, and the real experiment starts in September. And when that experiment kicks in, we'll be sampling DNA from these giant ocean simulations, and we'll be simulating a future acidified ocean, and then looking at how the acidification process affects the expression of a particular suite of genes that participate in this very complex biogeochemical cycle, which Steve Archer can answer all questions about. He's in the back. Okay, and then most recently, I'm getting to the end here. This is one of my newest projects. It's a really small collaboration with folks up at Colby College, and we're studying this organism. It forms these giant dandelion-like puffballs in freshwater lakes. It's called Gliotrichia. It's a cyanobacterium, and the reason that we're interested in it is that it blooms in these oligotrophic, low-nutrient lakes all throughout the Northeast, but it produces microcystins, which are a particularly nasty class of algal toxins. These toxins are what closed Toledo's water supply last summer, and I believe they're having a similar problem this year. So 500,000 people were without water for a week in Toledo last year because of this toxin. And so we're also developing the capacity at the lab to measure this toxin. Surprisingly, maybe it's because these things are enormous. They're a couple millimeters across when they form colonies. But nobody has done any genetic work on these organisms here in Maine. So one of the students in my lab, Becca in the back, can answer all the questions about Gliotrichia. She sequenced some of the produced some of the first sequences for this organism that will go into a national database and put Maine on the map as a location for where there's genetic information and ecological information for this increasingly important class of organisms. And again, maybe it's because they're enormous that people haven't developed a quantitative PCR assay, but we don't know what happens when they're not in these giant colonies. At some point in their life cycle, they're just individual cells laying on the sediment overwintering. Okay, so getting to the demonstration part here. If you were here at the very beginning of the talk, you probably noticed this little speaker-looking device that's up front. So this is my setup back at the lab in East Boothbay. Quantitative PCR machine hooked up to a computer. It has the ability to cycle 96 reactions at a time, take a picture of it. But it's not the kind of system that I would really dare move many times in its functional life. It's a very sensitive piece of equipment, not designed to go in the field. If you bump it too hard, you have to recalibrate it. So about a year ago, I started getting interested. I started seeing ads and press releases on the newest generation of DNA thermal cyclers. And then back in May, there was an article published in Nature about PCR technology coming out of the lab and into our everyday lives. So this is something I've been thinking about for a year, but suddenly the technology just really exploded. So I read the article, and the same day I read the article, I actually contacted one of these startup companies down in Philadelphia. It's a little company called BioMeme. They've been around for 3 years. And what they've done, I think, is really exciting. And this is not many people have actually even seen this device yet. It's still in the prototype stages. But you'll recognize that, let me just make a switch over here. Okay, you'll recognize, oops, let me check one thing. There it is. Okay. So you'll recognize that it has an iPhone plugged into it. So for the past 57 minutes, I've been amplifying DNA right here up on the stage. It's plugged in, but I could unplug it, although I didn't dare for this demonstration. And it can run on battery power for 2 or 3 hours. So this is the kind of device you could take out into the field. You could take it, a lifeguard could have it at the beach. You could take a daily water sample. When I lived out in Southern California, the beaches were frequently posted because you had 14 million people living in 40 square miles. And you just had water quality issues. But typically when a beach was posted, that meant that it was tested 2 or 3 days earlier, and you'd probably already been swimming in, wallowing in it. So what's the point? This kind of technology, you could run out to the beach, you could take a sample, and 2 hours later, you could have an answer, especially if it was done right at the lifeguard station. So so the reaction that's been going for the last 58 minutes, it's on cycle number 33 of 35. So if people can stick around for another 10 minutes or so, you'll see the results. But I've been running an assay for the red tide organism, Alexandrium. So I thought that this was an interesting organism because it's something that affects everybody who comes to Maine at some point through shellfish closures. And hopefully it won't affect you enough to send you to the hospital. But it's a very potent toxin. It's something we have to take really seriously. And there's a huge amount of work going on now. And DMR has been doing a great job at keeping Maine shell fisheries safe for us. But they're also quite interested in using some of this next generation technology out in the field, or if not, just back at the lab for these incredibly rapid assays. Another area where we see some application is in the aquaculture industry. Right now, there's a lot of interest in bacterial pathogens that one might pick up from eating raw or undercooked shellfish. And so imagine you could do a test to guarantee that a particular batch of shellfish was kept at the right temperature, was not going to make anybody sick. And it's the kind of test that even theoretically, seafood dealerships or even restaurants could probably do and will be able to do in the not too distant future. Sure. I predict that they will. They've only validated a few different PCR assays to date. The technology is sort of just at the point of switching over, but it's something that they're definitely interested in.
Would you review what PCR stands for?
Oh, yeah. It might have been on one of the slides.
It was. It was a long time ago.
Sorry? So the question was, what does PCR stand for? Polymerase chain reaction. Okay, so it's the fact that you set up this reaction, you walk away, and a couple hours later, you've got 1 million times the amount of DNA that you started with. Question?
I missed something at the beginning of this. You have an unknown sample that you want to, if it has certain DNA in it.
That's right.
You add to that markers that when carried through this reaction amplify greatly, but don't amplify the other thing.
That's right. They would be specific for that.
For the test you want to do, you need a marker, a chemical of some kind.
It's actually I typically target a gene that's called the ribosomal RNA gene. Every living organism has ribosomes, makes proteins. It's universally, it's a highly conservative gene. So we can find regions, we could line up the DNA from E. coli with the same gene from humans and find regions that are 100% identical for E. coli and humans. Over many, not just single base pairs, but like many tens of base pairs. I was actually this is a little tangential, but when I was preparing this talk, I read a statistic that our genome to genome, we're about 97% similar to chimpanzees, but we're 50% similar to bananas. So the fact that there's so much evolutionary conservationism you can find these primers that will work with a huge range of organisms. Does that answer your question?
Maybe.
Okay. Okay, so it looks like we're on the last cycle of this PCR. Oh, sorry.
Can Bigelow benefit commercially from doing these assays?
So the question was, can we benefit commercially? Can the lab benefit commercially? So we've actually signed a non-disclosure with these guys to help them develop assays because they're not going to go out. They're based in Philadelphia at a biotech incubator, and they don't have access to the kind of samples we have. We already are using this technology in our labs on a day-to-day basis. So what they really want help with is figuring out the new markets for the technology. And they have a very interesting policy, which they just put it right out there on the web, that if you help them develop an assay, there could be, if it gets adopted, there could be royalty sharing on kits. But it's also designed, okay, it just finished. They're also, I think, very much committed to the idea of democratization of this technology. And they're really, the guy I first talked to said that he thought that they would probably start selling these devices for about $1,000, but that his business partners want to get it down to about $250. So it's basically the price of a cell phone plus a little device, and you can do this at home. And in fact, the next version of this technology, they said that the iPhone will be decoupled from the thermal cycler, and you'll just run it right out of your pocket, and you'll be able to get the results, will be Bluetoothed back to you. Okay, so the reaction just finished. I'm not sure we can see it very well. It's a little bit too bright, so you may have to come up here to take a look, but did you see it? Oh, there it is, right there. Okay, so there's two. So the machine, the limitation of the machine is that it can only amplify 3 samples at a time right now. I believe a future version of the machine will be able to do more. But what I ran for this little test was 2 different species of Alexandrium plus a negative control. And you can see that the 2 positive controls worked very well, and the negative control remained negative over the different cycles. So basically it's not, it doesn't have the capacity to run the DNA standards, so I can't tell you how many Alexandrium genes were in that original sample, but it's clearly a positive. And for, in the case of pathogen detection, usually just knowing that 1 is there is important. So a plus minus type of test is very instructive. Yep.
You may have told us, and I may have missed it, but where is this machine being made?
So the question is, where's the machine being made? The tech startup is down in Philadelphia. They actually started out in Albuquerque, but Philadelphia had a very progressive program in place to attract smart young people to their city, and they gave them incentives to move from Albuquerque to set up right in Philly, and so now they're enjoying this biotech incubator. So this is something that actually Jose Antonio in the back and I went down to Brunswick Landing about a week or 2 ago because Maine has just set up a tech incubator down on the old Navy base. It's called Tech Place. They're actually getting more applications than they have room for. They're trying to find the best fit of a group of people. They envision by this fall of having a wet lab. So a company like Biomeme, maybe we can steal Biomeme from Philadelphia and get them up in Maine and create a really good partnership. Becky.
How much did Dave Karen's qPCR machine cost back in 2000?
Oh, let's see. So our former lab manager from USC happens to be in the audience, having moved from LA. Everyone from LA is moving to Maine. We're really good drivers. But actually, no. So the original technology that we bought back in 2002 in Dave's lab was $45,000. And again, they envisioned selling this little device for less than 500, say. I mean, even the one that I bought when I came to Bigelow was probably half that cost of the original one that we worked with. So it's just, stepping down and down and down. Part of this is that some of the DNA technology is coming off patent. It's been around for 30 years. So it's being released to the general public. And we were paying premiums for those patent protection, rightfully so. Yep.
Okay, I'm still confused. For this to work, you have to have this other, I don't know if reagent is the right word, this other chemical to add to it.
Yeah.
What's the source of those, and how do you distinguish those?
Well, we buy those off the shelf generally from biotech companies.
So they are standard?
They're a standard thing. In fact, Biomeme is also working on ways to provide tubes, reaction tubes that you could buy and stock in your kitchen that would have dehydrated forms of this chemistry. So all you'd have to do is add water or DNA.
Why do they make that chemistry now for this reaction?
Why do they? Yes, they make it.
Why do people make that for this? The thing you're excited about is scaling it down to a small device.
Right.
But the actual technology you're describing has been going on, just bigger devices and more expensive.
That's right. And so they want to make the whole technology available so that you don't have to go out to, the Thermo Fishers and pay $1 per reaction.
So if I wanted to test some shellfish for something, I would have to get a jar of something, add it to this sample.
That's right.
Wait for an hour, and I have my answer.
That's right. And now you know how to edit genomes and find those gene targets. So you're all set. Sure.
Pete, that one slide with that big round thing that's found in the New England lakes.
Yep, gliotrichia. Glia.
Is it found anywhere else on the planet?
It is. In fact, Becca, who's sitting right next to you, can tell you where else it's found. Because we were really excited. We generated the first sequences of gliotrichia from, I think, anywhere in the Northeast. You were number one right there. And when she compared it to the databases, it was from Finland or somewhere in the Scandinavian countries. And our gliotrichia is about 98 to 99% similar for this particular region of gene that we've been able to sequence so far.
Have we figured out how to combat it?
No, because I don't think we really understand its basic biology yet. We're actually going to a meeting at Bates Thursday, because there's a researcher who's been working on Lake Auburn and some of the other Belgrade lakes. There's a group between Bates and Dartmouth and Colby that have been working on this organism, but just not the genetic aspects of it. It's found throughout most of the freshwater lakes. If anybody's really interested in it, I can send you a paper on it.
I've actually seen that cell in the water down in Panther.
Yes.
Southern Maine. And it looks like snow in the water.
Yeah.
It
floats up to apparently it overwinters
down
in
in the
sediment.
wind
Yeah.
very easily as it moves that way. But they look like little.
It forms a surface, I believe it's buoyant, it's active, somewhere here. It's a really interesting organism. The other thing about it is that it's a this mesh of cells, and when we've stained it with DNA-based stains, you can see hundreds, if not thousands, of other microbes that are sort of living in those interstitial spaces. And when we first tried sequencing it, we picked out 20 of these huge colonies, and we got the DNA sequence from something else. So, it's apparently it's a good home for other microbes. Okay, I'm happy to entertain other questions if you want to come up and talk, but otherwise, thanks for coming out, and